So, in last session, we retrieved the 25 IDs. Okay, and we have added them into search box, and we observed this network. Is it? I think it is not shared. Share screen. So, are you able to see this? Please raise hand if you see. Okay. Okay, then. When we see this network, there are the different components we just able to see here, different colors of the line. First, I will talk about this network. So this is the node, and that is what entry we have added there. And this is the name of that particular gene. And then it has a connection with this. And there are the three colored lines which are there, that black color, magenta color, and yellow color line. So we will discuss one by one. So this is the overall network what we got. So now when you just go in the viewer, so you just able to check the multiple options. And here the network what we are observing now. So suppose you are interested to know the co-occurred genes. So out of 25, how many co-occurred genes? So if you click here, and then you will see that co-occurrence. So out of this, we know that gene co-occurrence, which is also useful to know that particular proteins are interacting with each other. So these are the multiple species you just able to see here, and what is the condition of that gene. So these are the 25 genes what we have uh, added, and out of this gene, that which are co-occurred, we just able to see. So the color codes which are shown here, that is they indicate that they are co-occurred in the particular organism. So if you see, the bacteria is here, eukarya is here, archaea is here. So in the eukarya, you able to see the dark colors are there, and this dark color indicates that these particular genes are co-expressed or co-occurred in the organism. So this is the one type of information you just able to get here, and you will see the tree, uh, phylogenetic tree like structure at the left side, where you can able to see the number of taxa and protobacteria, means what is the condition in other organism and so on. When you click on the plus sign, it will expand, and then you just able to see. So this is the co-occurrence thing, whose co-occurrence pattern across the genome. Then if you see that experiments, so when you click on the experiments, that how many genes or the nodes which are there, they are detected by a particular techniques, and they are showing that they are interacting with each other. So you will see that protein-protein interaction by co-immunoprecipitation is there. Then in vitro in vivo assay is there, and they are listed here, that mpk2, k7, k1, k2. So these are detected by this co-immunoprecipitation. So these are the list of proteins which are given here, and the relevant information. So which protein means, we are talking about how this network is drawn. Okay, so how this network is drawn, and this network is drawn based on which information. So this is the experimental information which is available for this network. Then next of that, you able to observe the co-expression, means as we told that if the particular two genes are expressed at one time, expressed, and it is observed in that organism, so then you can say that these genes are linked on. So suppose you will see here that dot is there, so this particular genes are co-expressed, and you just able to, when you click on that, then you get the details. So this is the based on the uh, experimental data, co-expression data, that is RNA. So you will see the co-expression observed in CKS1B and CKS2. So these two components that is the and score is 0.799 and 0.741. This is the co-expression data which is used to derive this particular network. So then databases are there. So you will get the information from which means you will get the details about the databases or experimental details from which the information is taken and drawn this network. So this is again which data sets are used, that Kaposi sarcoma KIC database is used to fetch as well. So this is some and neighbors. So as we say, the operons concept is there. So what is the gene neighbor? So the group of genes are observed in each other. I think this is an inactive one. And also this is not active. I think that data is not fetched. And text mining is there. So the text mining is one of the uh, um, uh, one of the component. So you will see this in silico method, means from where again the information is taken to draw a network. So this is the things what we did. This is the PubMed literature, means what literature is referred to show the network. So this is about the network, how the network is drawn. So if you have any question, you can raise the hands. Otherwise, I will move for next legions. Why? I think attendees are only 23 are there. The rest of not there. I don't know. So next of that, if you see this, the legions which are there, the legend means what it is shown on the top of your network. So here you can modify the content, means if you want to modify the node and everything, you can able to do this. Not modify, you will able to see what is the legend for this. First, that is the color nodes, white nodes, what that is they are, if the color nodes are there, they are first shell interactions, and white nodes are there, they are second shell interaction. Then empty node are there. In some nodes you will see the structure is there, and some node it is empty, sorry. So it is empty. So this empty node means whose structure is not available. Okay. Then field node, that is 3D structure is available. Then what we are talking about, that lines indicate that green, yellow, or so. So you will see that this line indicate what? So it is a cyan color line or magenta color line or green color line or red color line. So what type of data support to draw that edge? So that edge is colored by the specific color because it is taken from this particular data set. So the curated database is used for, and it is shown as a cyan color line. Experimental data is there, and you can show that is magenta color line and so on. So these are the edges which are shown here, which is drawn based on the what type of interactions means edges are drawn based on from which information they are drawn. That is either there is or which evidence, either it is the text evidence, either it is experimental evidence or so on. And you just able to see this. So this is the legends and information about each entry is given here. So what is the information of this? Then you can able to see the settings. So you can change the component here. So I will mm, see that the full string network you want or physical network you want, you can adjust it. That as we say that genetic regulations and so on. And next thing, you don't want, this is the evidence based, you want only confidence, that is 0.8 or 7 or so. If you just click here and you can update, then confidence level edges are drawn. So the thick line indicates that the high level of confidence, thin line indicates the very less confidence. So you can remove the evidence to the confidence. Again, if you are interested to only the interactions of uh, text mining, or if you are interested only the evidence source is experiment, then you can check this and remove this. Then you will only get the network which based on the experimental evidence. Then you can change the confidence level, high confidence, highest confidence, and so on. And you can update it. So when you update, only high confidence level network will remain here. You will see here. And these are the nodes which are initially are in linkage, but it is not this. So again, if you want to increase the interactions, that first shell, second shell interaction, that uh, two or more routines which are means 5 interaction, and so you can increase means additional interaction. So I will increase here 5 and I will update, then additional 5 proteins which are interacting with the particular node is displayed and reached in that, means added in that. And then next of that second shell, if you want, you can do. Then what type of image you want, interactive svg or static png, you can see. And display modification, if you don't want a structure, disable structure preview and you update, the structure which are present inside will go away. And if you not want to disconnected nodes, those who are, suppose this CDCP and CLK is there. So if you just click here and update, so the disconnected nodes will be hidden. Okay, so they will go out, as well as show protein input protein name. So the input protein name is displayed here. Otherwise, it will present on the node itself. So if you just able to see this, so this is the modification you can do in the graphics, what you display on the string. You have any question? If you have any question, you can ask or raise hand. I will unmute you. I think you understand everything. So next of that, the analysis part is there, and which is a important one. So here you will see that the number of nodes, how many are there. So this is a listed here. Then how many number of edges are there? Average node, that is 4.53 degree. And average local clustering coefficient 0.61. Expected number of edges, that is 17. And PPI enrichment p-value, that is less than 1.16. So if you see that, so this is the analysis part which show you the network statistics. And then you just able to see the enrichment. So enrichment is one of the important component for network, because suppose you have identified a protein and you don't know the function of that protein. Suppose microbiology. What is the enrichment media? What you enrichment you did in microbiology, isn't it? Or anyone wish to give the answer? What is the enrichment? Yes. Mm, when someone is raised and Chitij. Yes.
It is used to increase the density of microorganisms.
Means what we doing there in the enrichment?
We are culturing more, we are
No.
amplifying.
What we do, we actually adding some additional components. Okay, Ishrat. Yes,
Yes.
Ishrat, you can speak. Yes, I have unmuted you. I think it is not. Please wait. Uh, yes.
Hello, sir.
Yes.
Hello, sir. Can you hear me?
Yes.
Enrichment media is the media that allows the growth of only particular type of microorganisms.
Okay. Okay. So what you do in that from normal? What is different in that?
We are adding micro and micronutrients.
Mhm.
For the growth of, say, fastidious microorganisms to grow nutritionally, you need to exact the particular type of microorganisms. So you add a particular nutrient which will allow the growth of only that type of microorganism.
Okay. Okay. So what we do actually that we add a particular type of microorganism in that, not nutrient in that, and then the growth of a particular organism is increased. Not all. Okay. So we are enriching with the certain nutrients. Okay. So if we see that the same concept is used here in functional enrichment. Suppose you are going with a list of protein, then you submitted the list, and then um, you have list, but you don't know what is the function. So actually, this protein is involved in what? So the functional enrichment is done here. That is based on the similarity of that sequences of the protein characteristic. And now you will see that the function prediction is done by enriching. And there are the some values you will see that there are the functional enrichment, how biological process means how many proteins are involved in the biological process, how many are involved in the molecular function, that is how many are act as a cellular component, how many are as a reference publication. So this enrichment is done, and you able to know the which proteins are expressed, means take part in the biological process, and in that cellular response to sorbitol, regulation of Golgi inheritance, Bergman glial cell differentiation, or mitogen activated protein kinase. So if you see this one, now then if you click here, then you able to see this red color spot, and cellular response to sorbitol. So which are the proteins which are respond to the cellular? So these are the MAP2K6 and MAP2K4, which are give the response to sorbitol. Regulation to Golgi inheritance. So these are the two proteins which are given a Golgi. So these are involved in, suppose, Janus kinase activity. So if you click here, so everyone thinks how a colored, and you will see that MAP kinase have two now. It doing two activity. One is that, one is other, that yellow and magenta. That's sorbitol as well as JN kinase. So this functional enrichment will give you additional information that these particular proteins are involved in which biological processes, or what is the function, or what is the cellular component, and that is the mostly the protein interaction networks used to predict the functions, because in the bulk of protein you have identified from any bacteria, you don't know what is the functions, and using functional enrichment is identified that this protein might be this function, and according to the work is forwarded. So this is the ERK disease. So so you will see that so ERK. So these are the two. This is the functional enrichment analysis shown here, and this is at the hematologic cancer. If you see that dark green, so these are the involved in the hematology. So all type of enrichment is available here, and you just able to use. Okay, so this is the functional enrichment analysis you can do here with the help of a string. So next of that, so many, we filter out the microbes. Okay, more nutrients. So if you see that this functional enrichment is there, then we just brief idea that which proteins are involved in it, and we can act accordingly. So so this is one task is there. So someone is raised hand. Yes.
Sir, can I interrupt in between or at the end I will
No,
ask?
no, you can ask.
Sir, why then it is named as false discovery rate under the title of false discovery?
So this is the FDR value for gene expression. So this is the FDR values for gene expression. Means what we can say that any gene is expressed, then it is converted expression calculated in log value, and then it is converted into FDR value.
Achha.
FDR value indicate that what is the contribution of that particular protein in that cellular response to sorbitol. So if the FDR value is near to what we can say 0 or down 0, they are better one. And also you just able to see the strength and everything which is here. So with the help of this functional enrichment, you able to identify what
In short, it is like a e-value or confidence value, right?
yes,
Oh,
yes. And you just able to identify what is the functions of this particular.
all right, sir. Thank
Yes.
you.
So when this is the functional enrichment analysis, what we did, and you able to decipher. But you are now you think that why we do this and that. When you just go for any research problem, then you understand what is the use of this. Because if those who are working in the microbiology, those who are working in the cancerous cell, those who are working in different cell or tissue culture, or they are working on the rDNA or expression and so on, then they understand why the use of this, because large amount of the gene and protein product you will get. and it is difficult to handle. So then next of that, this is the export option. So you can export this network in bitmap image, high resolution bitmap, or you can export in XML summary or network coordinate or routine sequence and so on. And you can able to see this interaction as well, that ABL1, ABL2, what type of interactions and what is there. So this is the export information. Then you can able to do the clustering as well. So no clustering is there, K-mean and MCL clustering. So these are the multiple algorithms which are there. So which is used for performing the clustering analysis. So if you did the clustering, then you will able to get the cluster. So dotted lines, so this is the single clusters and they are colored in the different colors. So red color, that is a single cluster, and dotted lines, that is the another one which is joining. So this is the individual clusters you able to get. And based on that also, you able to predict some more thing. As well as MCL clustering, they are different algorithms are there, and cluster 1, cluster 2, cluster 3, you will see that how many genes are there in 1, 2, 3. So more, if you want to add more nodes, then you can click here, then nodes will going to add. And if you click on less, then more nodes will going to reduce. Means what we do in the settings here, that what we can say here, that first shell and second shell. So that things we can do from here. So now if you have any question, you can ask. Now we have to move for next component, that is cytoscape. If you have any question, any queries, you can ask on this. No question. That's so now there are the no lot of things we able to do here in the cytoscape, in the string. Further topological analysis of this particular network we need to do with cytoscape. So I will show some slides which are there. So this is what we so after searching a network, then I have drawn the network and moved towards the cytoscape. Then our interest is to identify the particular nodes which are key play. So we know that because we are interested to identify a target. For example, suppose you are started with your pathogenic genes, pathogen genes or proteins. Then you are interested to identify a target, and which is the best target, that which has a key role in metabolic activity of that. Okay. So this is mm, the first analysis we do, that is the degree, means there are the different parameters in the first previous slides. I have shown you what is the hub node, what is the degree, what is the centrality measures, what is the average centrality, what is the degree. So this degree means there is a number of connection of a particular node with other protein, okay, or number of links. And with the help of these uh, interactions, we able to identify the hub. So here at the left side, you will see that the degree is going to increase in this way. So this is the degree, this is the, and when we calculated it against the between the centrality and degree, you just able to see these are the orange color spots. So this orange color spot means each node is there, and the higher the degree, the better the node is there. So uh, and we just set these parameters in the cytosol to identify which are the hub proteins. So the higher the degree means it is the criteria you have to decide that what is the average degree, and above that you can decide the criteria, that is above average those are there, we can say that these are higher, means better, and so on, because they are key players, you can say. And when you just do this, then you able to see this is the normal string network which is there. And after that, when we just filter them or change the style based on the degree, then you will see these are the proteins which are key. So if you see this other one, they have no any uh, means effect on the overall, means might be they are not participated actively in this particular interactions. So one minute, please give me. Hello. Hello. So if you see this, so this is the hub node what we see here, and when you look into the network, the sizes of every node is similar here. But here, the size of nodes is increased, some node size is decreased. So the increased node size, it indicates that these protein are main players in the pathway. Okay, these proteins are main players in the particular path. This is the uh, selection of a protein, means when you just do this, when you are interested to identify a target protein from a pathogen, you take whole proteins from the pathogen and you can identify the targets. If you are interested to, means it is a change from means what type of research you want, means you are doing. Based on that, suppose you are interested to know the tolerated plants, then you can see that which are the genes or proteins which are involved to increase the tolerance of that plant, or disease, which are the main protein in the bacteria, also in production, also a particular production you want to see, you can do this. So Then the clustering is done at, or these are the different types of the clustering which is there. So encode clusters are there. So this is a cluster what we generated on this network. So these are two clusters which are there. That is one cluster and second. And I hope that similarly we will. This is a different genes, and so these are the two clusters what we observe using M code. And next of that, hub protein means you should not confirm. Means first time when you did with the degree and you decided that yes, this is the main hub protein, because you should not just means decide on a single analysis. Means you perform the hub node first time in the cytoscape and you say yes, these are the, this is not the way. That you need to confirm the same thing by multiple times by using multiple, because in this in silico work there are each algorithm have their own advantages and disadvantages. So you need to perform this tasks, okay, this task multiple times using different algorithm or different analysis tool. So here the CytoHub, which is a popular tool, which is also used for identification of hub node. So that red color or dark red color, which is in said that it is a main, and then there is a yellow color, which is again not a good one. Means it is based on the degree the hub node is calculated, and MCC is used to calculate. So this is the network analysis what we will going to perform now. So if okay, we will start the cytoscape. If you have any question, you can ask. Sandra? Sorry. So after this network, someone have query? So if you see this, the export option is there. First, I will talk with the cytoscape. Is it cytoscape visible to you all? Visible to all? You can simply raise hand. Okay. So this is the freeware which is available and you can download it on your PC, and any means network related or some other features are also there. So and additional tools which is required. So if you see that this cytoscape platform will useful for drawing a network in artistic way, as well as it is helpful to analyze the network. So you will see that in app there is app manager, and you will see that the install app, currently installed app, and check for updates. So you able to see how many apps are there. There are 243 already apps for collections. So that much number of apps you can install in the, it's multiple apps are there. So please note down what apps are required actually. So you have to select and you have to click on the install. Then app automatically install. You can check from here that which apps are already, check for updates. So these components you can able to. So what we can say uh, that three other tools are you need to update in this, that M code is there, then Clugo is there, and CytoHubba is there. So and network analyzer. So this three four tools you need to install in the cytoscape if you are performing the work what I am doing. Okay. So this is the cytoscape, and I will show you the thing component means how to perform it. So as we seen the export button here, so the export button, if cytoscape is already installed, then you will get this sent to cytoscape. Directly it will forward. If you don't have such a button, means you're not able to observe this button, then simply you select it, the string protein query. You have to first list. I have first name list, and then you need to select what organism as normal what we seen. So Homo sapien is already there, and you click on the green. And when you click, so 25 identifiers are there. 25 identifiers will load. Yes.
Sir, really sorry to interrupt, but there was some network issue in between. I lost. Can you just tell me from the beginning 2 steps that we did?
But if you have
You can just
installed,
huh.
yes, if you have cytoscape,
Yes.
then install, then
Mhm.
open the cytoscape, and
Yes. Huh.
then you have to go in export option in
Yes.
the cytoscape. Here you will see send the network to cytoscape.
Okay. Okay. Huh.
If you're not able to see that link, it is
Huh.
possible that sometimes you're not able to see that link.
Huh.
So at that time, you able to go here, that network, and here. So here multiple databases are there. So you have to select string and put your protein query. Okay, you have to
Okay.
paste your protein and you click on this option to select the organism. Suppose it is Homo sapien, it is already default. And you have to confidence level how much you want and additional interactions and enrichment data you have to click, and you
Okay, sir.
click on the search option.
Perfect.
Then
Thanks a lot.
automatically the network will drawn.
Okay.
This
Perfect, sir.
network,
Understood.
so the network is drawn here. So now you just able to see. I hope you able to see this. Yeah, be open. So you able to see this network now. Okay. So this network which is drawn here. So here that the string setting is there. Okay, the string app, if you install, then everything is there in the string. So how to open that app? You have to go in the app, then you will see the string here and expand the network or open that. This panel will open. Okay, so in this panel, you can modify the display style. If you see the glass bubble are there, string networks are there, names, and you can able to see are you able to see this now? And then you, I think. Okay, so this is the network what we see here. So when you remove this, this is for a single node which are not connected. It is removed. You so this is all the additional things which are there. So functional enrichment, select a node from initial query or retrieve enrichment and cluster. These options are also available in this string. So when you execute this option, then automatically it is applied on it. Then tissue filter, suppose you want to know that you are interested only that which proteins are expressed in the stomach. Okay, you are interested to know which proteins are expressed in, I will just drag it to the left side. So these are the values which are there. So you are able to view it properly. Means you are working on what we can say. Can you please send a download link for these tools? Yes, yes. I will send it. So if you see that the protein which are present in the means out of this 25 tissue filter, you are interested only in the protein present in the blood. You are not interested in bone or bone marrow and so on, or eye. So you can use this to filter them, that this option. You will see that here that protein present in the blood, you will see that is going to filter. So that type of tissue filter you can use here, okay, for filtering your data. So if you are interested, why these options are here? Because a lot of this is 25 proteins we have taken. So it is we not able to see, it is difficult. But if you just go for large number of protein IDs, large means 1,000 or 2,000 or 1,000, then it is quite difficult to. So this filtering option you need to use, and then a compartment option you able to see, that is the which proteins you are interested only in the extracellular. not extracellular, the protein which is expressed in the nucleus and so on. So you can filter them. So you are interested to know only the protein which are present in the nucleus. So you can able to filter them. Likewise, you just increase and decrease the bar, and you able to see the protein which are expressed in Golgi or expressed in cytosol, expressed in cytoskeleton. And so when you select the particular node, you will see the information of this particular node. The, the information of this particular. So this is one thing. You have any question about this? So this is the string analysis. So what we talk about. So this is the string analysis which is there. Now again, what enrichment we observed, I will remove that single node. Enrichment, what we observed in a string, same enrichment you can able to see here, and the different multiple colors are there. So that is the, and you can string enrichment table is there, and you can able to filter that well. So you can able to filter that using this option what you want filter in the table, and you able to see the details that what are the go-molecular function protein kinase, which are the proteins. So this is the enrichment. Then network table you will see here, that is what network table, and node table and ages. So this is the node table what you able to see here, that is the node which is available the particular network. Then age tables are there, so how many ages and how they are interacting with. So you can check the table as well if you are interested, and if you remove this. So this is the string analysis for a particular network. Means string modification you can do. When you sufficiently do this enrich, means initially when you use this option and perfectly do the activate the enrichment data and so on, then you will get a bit. Then I will also wish to show you, these are some options which are there. This is the grid, or this is, you can take it away. So that particular window, network window, you can take it away from this place. Then you can use this to remove the nodes that you want to flip and something, and able to do it with this. You can able to add the ages and anything you want to select from the particular area you can do, and you can toggle the text and everything you want to move, then you can able to do. So this is the text and everything you can able to do here. Okay, so this node movement and everything is possible here. Okay, so we will not go for this, and this is the some snapshot you able to see that how actual network is. So this is the string network which we pulled here. And now we will go for analysis. Okay. So what further analysis, that is called a topological analysis, is there? And a topological analysis. So you have to go in tools and you click on analyze a network, analyze as a directed graph, and say okay. So when you say okay, then you will see again same information is there. That is the string network, that number of nodes 25, number of edges 70, average number of neighbors, network diameter, radius, characteristic path. These are the topological analysis of the network. And you just able to what I shown the two graphs about the nodes. You can draw this from node degree distribution. I don't think. Sometime it gives a problem. That's why. So when you click on this, I don't know why it is saying. There is that graph what I have shown here. This one, that degree distribution and so on. So this one will display that one is no degree distribution and between is centrality. Sometime it is give a problem, but when you close the system and again start, then it will work. Okay. So, but the betweenness centrality, so this is the nodes and everything is there. So you will need to decide the degrees and so on with the help of these graphs. Okay, that betweenness degree, or you can decide in the table. You can get that information in the table as a network table, where you just get the detail about the enrichment and so on. So the, the table is also there to analyze this. Then you have to go for a style. So the style which give you the multiple options to change color, color of nodes, and so on. Colors, label size, and everything you can able to change with the help of styles as well as border width and so on. But what I shown you that hub node and so on. So this is you just able to check here that height and width. So height and width means whose degree is more than that particular node is hub node we call. So you have to go in the height. Now, why we have to go in the height? Because the height of these nodes and everything is same. Okay. So we have to decide the height based on the degree. So you have to go for a degree here, and then you just go for continuous mapping. So when you go for continuous mapping, then you will see the graph. Okay. You have to double click here, and what it shows that 0 to here 10 to 30, and here what it shown actually degrees values are present from 0 to 14 means total. So you have to click here, that is it is at 10, and it is already there. And here you have to set 140, because so we have set the value 140 here. Okay, and why? Because you see this degree values for this network, that 0 to 14, that normalization. We have did normalize for height. That is according to 10. 10 will go to 100 value, and 14 value will go to 140. So you have to say okay here. Then you have to go for width, and the width as well. You have to select a degree, because now nodes are looking stretched. Okay, where is it? So yes, here is the degree. And again, you have to go for continuous mapping. You have to see that graph, continuous mapping graph. Yes. So here also, you have to go for 10. If it is already there, you have to go for 10. You can say, okay, you have to double click here. Okay. And here you set 140. Okay. So you will see that this is okay. Now, out of all nodes, you able to see that the some nodes are increased in size and some nodes are normal. And we have increased the size based on degree. So with the help of layout, we can able to expand them. Okay. And you can rotate here. So what you expand here, it is height or width. So you able. So now we identify that these are the hub node, that is EGFR out of 25. So you will see this is the another one, 3 protein, and this is the other protein here. So this MPK14, EGFR, JK1. So these are the main players, MPK, EGFR, out of all 25. And Jack. So because then size of this are reduced, because why this degree means there are the links between the author. They are linked with almost all these protein which are present in the network, and that's why their role is more in this particular network, and that's why they are highlighted. Means increased in size. So if anyone have question, you can ask. And here you can also do one thing, that styles you can change, what network style you want. You can change, and suppose biopacks or string network style is here, you can use any one style which is available, and color, label, font size, and everything you can able to change. That is the label font size here. So I will label 20. I will do that. So it will change 20. A lot of things are there in the style. Okay. And then the filter option is there. So filter option is there also. So if you want to filter something, that is column filter, degree filter, you don't want to show the hub nodes and so on. You want to show only the protein having higher degree. So when you just do that, and you have to set and apply, then automatically this is 0 to 14. So suppose you have stretched it. In, out, out. 0 to 14. So if we apply, you will see. So that degree is going to apply here, and here you able to see that is how many nodes are there. If you say the select, then it will select it. You will see here 2 to 7, 1 to 7, or here if I change 14, likewise selected. If that particular, you will see that. So that particular component is going to select it, and then it is displayed. So you just able to see this network and filter. So any questions? Can you give one example? There is a problem. Related to what? So suppose you are interested to know that I will give the example of pneumonia. Pneumonia pathogen is there, and you are interested to design a drug to kill that pathogen. And you have information of almost all the genes of pneumonia. And when you identifying a target, then you need to know that which is the main protein we have to hit, because suppose 200 proteins in your hand from the pneumonia bacteria and all are involved in the metabolic activity. You cannot design the drug for all 200s. You need to decide which is the main. So you just do this analysis, that is which is a key player, and then you will identify, yes, this is the protein which are a key player and which disturb overall metabolism. Why? Because it is interacting with almost multiple. So this is anything else? Sharwadi raised the hand. Yes.
Yes, sir. Sir, if we talked on the same line, means on the same example like pneumonia. So when we are, you said that we can perform this basic analysis. So if we have knowledge of about 20 proteins which are related to pneumonia, then any one out of it can be given as a query and we can go ahead?
Any one?
Out of those 20, if we have knowledge of 20 proteins which are involved in pneumonia pathogenesis.
Yes, yes.
Okay.
We can go now. We can go. Because suppose in string you see that there are the multiple search options are there.
Yes. Huh.
So you can give one protein and that particular protein interacting with which other protein you will see.
Okay.
And then you can do the enrichment, means even though you need to apply the logic here, means if you just added the protein, you got the network. Suppose
yes,
20 proteins are interacting with your protein, then you identify hub node. Okay, hub node. So hub node you identify, and you identify that one ABL protein is hub. Okay, it is main protein out of 20.
Okay. Huh.
So, but out of ABL2, if it is structural protein, listen, if it is protein present in cell wall, okay, then it is of no use. Okay, because you not able to, means if it is enzyme, then you can inhibit or design a inhibitor. If it is a structural protein and which is present in the membrane, you cannot design any drug to, and that protein will not interact because they have no any specific active sites and so on. Okay,
Yes, yes.
so further we need to enrichment, means only this identification is not end here.
Not sufficient.
we,
Yeah.
we need to confirm with 2-3 tools again, and again we need to do the functional enrichment, means what function that protein perform in the organism.
Okay.
And when we know the function, and if you identify, yes, that protein is involved in the gene expression. Okay, after enrichment analysis, then you specifically inhibit it, then automatically, and the bacteria will kill because of the drugs what you develop, because that protein is involved in the gene regulation, and it stops the expression of multiple genes, isn't it?
Yes, sir. Yes, understood. Thank you, sir.
So what I will do now, this is the filter option uh, which is there. Now I will show you the encode analysis. Again, that is it any specific clusters which are there? That there are the specific cluster, means a particular group of proteins are interacting with each other or so on. So you need to go on the app, which is I have already installed. So you click on the M. So mcode will ask for parameter, that whole network we need to consider, or selection what degree cutoff you can increase if your degree are more, and haircut is there, node score, and so on. So when you just click on analyze network, then you will able to see these clusters. So again, the score is important one. So the score, default score, that is 4 and above score is better. Okay, 4 and above score is there for the cluster, then that cluster is better. So out of that 26, I think how many nodes are there? So these two clusters what we identify, and you just able to see these clusters. Okay, so this is the cluster one, and I will do one thing. Cluster network, create cluster network. So I have created a cluster network. Okay. Similarly, for second one, I have created cluster network. Okay. So two networks I have created. So here you will see the original string network having 25 nodes and 70 edges. And here these are the clusters. So map kinase 26, 14, and so on. So these are this is one cluster whose score is better one, and this score is 3-1. It is not that much. So further, what we need to do, why we do these things are multiple times, because we should have confidence on what we are selecting. That multiple times we are doing this because we should have confidence on the particular protein. That protein is a key player in the metabolic activity of that organ. So here that cluster is, so two clusters are there with score you will. Again, if you confirm that, so this is the major cluster what we find, means we are, these are the proteins which are present as a hub, and same protein we observed in the hub node. So it is second level confirmation that is our, these hub nodes are hub proteins are important in the pathways of that organism. So then, 1 minute. If you have any question, you can ask. So these networks clusters are there. So again, this is a second level confirmation that what hub nodes we identified, they are main players, that EGFR, ABL, Jack, and then MAP kinase 1. So you can able to see MAP kinase 1, ABL, EGFR. They are come in the cluster with a high level of confidence. Next of that, again, we need to do the second level confirmation, that Cytohubba. Cytohubba is a tool which is used to determine the hub nodes. So it calculate the node score. Okay, so what I have selected, you select what you want. Either you want this cluster or either you want that particular string network. So I will select the cluster because what we said that already we have filtered them, and then I will select the cluster, and I will select the top uh, 25, that MCC degree. So again, this is the second level validation is there. So among them, what are the hub nodes? So 1, 2, 3, 4, 5, 6, 7, 9, 10. So the 10 is there, and this 10 I have submitted. So after submission of this, you able to see that these are the red colors are of node, and you're not able to see its names. So the names are not properly visible. So you have to go in the style. You check at what is the labels. So you can select the names, display names here, and it is automatically. So here again, we see that EGFR, that is a red color. So Cytohubba, if you see here, so you will see that the names are there. And this is 1133, means 11 means the ranking, that two are at the same rank. So that second ABL, Jack and EGFR at the same rank, means EGFR and Jack 1 and MAP kinase and ABL. So these are the key players out of this 10. And if you look into the previous network as well, if you see that our initial network, so the EGFR, ABL, Jack, and MAP kinase 14. So this is the play. Again, you can use the Cytohubba using multiple algorithms. So what I shown here, I have shown here the degree base. You can use the MCC, DMNC. These are the different parameters which are now you will see that bottleneck. So when I submit it with the bottleneck, the situation might be different. So you will see here MAC 14 is there, which is me. So map K14, if you see the initial network, that map K14, which is work as a bottleneck. So if you see that the particular cluster which is there, so this cluster is there, which is directly attached to the map K, and these secondary clusters which are directed from the map K. So that's why it is selected with the highest. The map K, Jack, EGFR again, but bottleneck map K14 have highest score and rest nothing. So you just able to confirm, means what type of means you just able to use the multiple algorithms which are there, means MCC, bottleneck, EC centrality, degree. So these are nothing but their interaction, their shortest path, and so on. So these values indicate the information about. This is the again what we confirm that. So EGFR, EPL, and MapKJack. So these are the main players in this particular. So So this is what we do with the cytohormone. So if you have any question, you can raise hands or I will move on. So, so I will open one more app, that is the Go. So Clugo is one of the functional enrichment app. So what we did, we know that this particular 10 proteins are involved in the means key players are there, and we are interested to know that what is the functions. We don't know what is the functions. So these 10 proteins are there. We don't know this. So we need to do the functional enrichment, and Clugopedia, so the app which is available. It is also freely available, but you need to do the registration for to the website, to fill the registration form, and after 6-7 days you will receive. But I think institutional mail ID is required. They not entertain Gmail or Reddit mail or yeah. So you need your institutional mail ID. You can freely download. You can download and install, but it will ask for a license. So you need to put the license key. They have. They already give the instruction how to update, and you just. So here the Clugo and Clupedia, which is there, and this Clugo and Clupedia, which is used for functional analysis, that genes and so on. So here, what organism you have to load that particular data set. So I have selected Homo sapien, and you will see that download new organism or refresh the organism. So this is the database update, because this is the annotation database having the information about then gene and everything information. So then what we will do, we will go for so it will ask for a automatic or accession, means any IDs they are asking. You can able to say IDs or cytoskeleton or seance. So what the information is there, they can ask for it. If you just network, then if you selected a file, then you have to add the genes. If you selected a network, then they will check the IDs which are there in the network, initial network of the system. So if you give the files, then I will give for file Jack1. At each line, ABL1, EGFR, to K6. I will put all this ABL2 because we are already finalized that these are the genes which are players. Then we will only tag ABL spiky map to K1. I don't know, it is added. Map to K1. So if you select the network, no issues, you can go for network and it is selected. Then visual style, it will ask for group or significant. Means if you want based on the significance or you want. Then what data sets, ontologies you want to follow, that is the biological process, then cellular components. So I will select the biological process and cellular components here. Means what the functions function of that particular listed. Then evidence, what evidence you will prefer, the experiment or other component. Then you will see that go category. So KEG category you want, KEG human diseases and so on. You just update it because a lot of things are there in this Plugo, and it need to decide based on what information about the genes required to user, and according to that the user select respective go term, respective evidences. Suppose you are relying on the experimental evidence, you can select the experimental evidences for that. Otherwise, you just able to means in silico you not rely on that, then you don't select that. Then the new organism data, if you want to add, you can add, and you have to network specific. I will put here somewhat global. Okay. So use go term fusion, means multiple terms are going to fuse. Then there is a tree interval. They just going to ask tree interval maximum 3, minimum 3, maximum 8, means particular tree which is going to drawn there. For example, the two genes are there, how much they are closer to each other, means 3 genes are there, then there is a distance, or maximum 8 are there. Then clusters are there. So how many clusters you want? Minimum 5 it is said. And then go term pathway. So the kappa score is one of the validity or validation parameter for identification of functional enrichment or annotation. So you keep it high if you have a lot of proteins, and then only few terms. Then statistical options which are there. These are the statistical options which is used. Bonoferini steps down, then PV corrections, mid P value. So this is again enrichment, depletion, what you want. So these are the parameters which are used for the this gene. And grouping option, fix random, test significance you want to see, kappa score, anything, means all these are what layout you want. So you can see logo layout, and you can start it. So only few proteins we have added. We will see that it will come or it will give the error. Okay. So you will see that the biological functions and this are coming. Okay. So what I will They are not going to say. Okay, only 4 or 5 are there. We can change them to the layout. So these are some annotation what was written here. Some detailed clue analysis. So lot of things you need to do here, that separations, and it is not easy task. Means you just use the clue, and everywhere you able to see that this particular color is given. Okay, so this green color is given, and other color is given, and now you think that how you come to know that this protein has this function, this particular protein has function. So this is the multiple activities which are shown here. So when you see this Clugo result, so in the Clugo result, you will able to see that a particular term. I will hide that minor terms. There is one option. So that smaller terms will go away and larger terms will remain as it is. There it is. Okay, we'll see. So that Go terms which are there, that gene ontology terms, so peptide transposil modifications and so on. So these are the some terms which are ideal. So activation of protein kinase activity and transmembrane receptor protein kinase activity. So this your proteins are splitted into two activity. Then if you just go in a particular node, you able to see that. So which are the proteins which performing this? Are the protein which are performing this act. So this is the details about your protein you will get. And if this is the clusters what you identify. So there is a two major clusters we have identified, that activation protein kinase activity, that is 69% are there in this, and transmembrane receptor activity is that. So if you read this, these are the additional terms. Okay, gene and term. So high level of confidence is here you can able to see, and there are the less map kinase activity, again sub activities which are listed here. Okay, that is what is this transmembrane receptor activity means, what is this, what is that, what is this. So these things are displayed here. Then you able to see that the cluster information is there, what score means, these parameters what you applied you will get here. And the Clupedia result, you just directed ages, undirected ages, you can able to see here, as well as you just able to change the this graphical representation as you want. So in relation to the kappa score, in relation to the interactions, or everything you can able to see here. Okay, so this is I am interested to remove that small member. And then label is that is it. So when you just take this particular component, okay, so do you identify that 10 genes which are able to do this, and you can change the display style here, that logo prefused force directed, you can apply, you can change it in swapping, means you can apply on your own what you want. So this is the style you can able to change here from this. And if you want to change the parameters, kappa score, everything, you just able to uh, change this component. Now what we did, network specificity, global, medium. If we say the global, then in general, if you say the detail here, if you see the detail here, then you will see what is going to change. So global layout. So detail means then the detail of that particular protein. And what proteins are present in that particular sorry group, you able to see. You will see this. The detail means again there are the detail enrichment is done, and multiple proteins are displayed. That is, which proteins are involved in interleukin 2 mediated signaling, which proteins are involved in uh, mRNA activation, which are in Bergman glial cell differentiation. So additional details you will get, and with the help of this, when you just do this, you able to know that which genes are there. Okay, you able to know that which genes are there in this particular cluster, because the gene list is there. In this particular go term, that particular gene, which genes are there, just able to identify, as well as so in this also you can able to see that how many terms are there and what are the different terms which are available. Then you can use this network and you can change the style what you want. If you want a label as per a particular thing, then you can terms, you can change. You are interested to know the associated gene, then you will see associated genes are going to display. Okay, so the associated gene values are there, or gene cluster if you want. So that labeling you just going to change. That particular labeling you just going to change with respect to. So you will see that what are the all other associated genes with this. Okay, so now you will see the ABL, GPF, MAP, KMAP. So you can decide that after functional enrichment, you can decide that which protein you need to target, because if you just convert into term, then uh, percent term, then you will get what is the function or ontology of that. And if you convert them into, if you get converted into term, and what is your interest, so you can check that what is the uh, function of that protein, and either when we block this protein, be able to kill that bacteria or not. So suppose specifically if you selected positive regulation of mRNA, that is the genes which are involved in the expression or which regulate the expression of bacteria, you can able to develop an inhibitor and which will. Okay, so if you have any question, you can ask. Question. Raise hand. And I will show you layout of the cytoscape. When you go in the network, this is the string network. Okay. So from here you can circular layout. All nodes. You will get like this. You can change it. Hierarchy, grid, stack और preferred मतलब original आएगा. Apply preferred layout. Not to interchange. Any question is there? Any question? Ask the question if you have any doubts. Any to score and everything you can layouts and everything is, and I will show you. So likewise you can able to do in this flipping and everything is, you can move the label. So this label you want to set aside, you can move it. This is a text add karna hai. You can add the text there. I think. Okay, any question is there? You have to raise hand and ask the question. Someone want how to download Cytoscape. So you have to go to this website. Here you will see download Cytoscape. I will paste in chat box. Have you understand anything? And again, I wish to say there are a lot of functions are there in this. There are the couple of apps which is available for a biological analysis, which is shown here. But if you see in the app, so you can able to see that how many 243 apps. And suppose you can load the structure from this import you will see that NDX file is there, network from file, network from URL, public database, table file, means you can import. So suppose URL is there, you can copy, I will copy, we will see. String URL, I have copied URL. We'll see if it is. Where is the error? The network without selecting a source target column. But it comes, I don't know. BioGrid और other tools का network आता है. So I hope you understand little bit and you're able to perform it on your own. That I have added that a particular link in that box, and you just use the string, follow the cytoscape. Any queries, anything you wish to ask, you can ask, because tomorrow the different session is there. I will be not any query, anything you want to ask, and you can. Because I think what we planned, it is finished, or what you not understand in the what we learned from morning. So you can ask on that as well. I hope you understand everything. But if we have Ayurvedic formula which constitute 3 phytoconstituents, we need to design network pharmacology for it. So what step follow? Because we need to do it. Okay, so if Twinkle Mishra, if it is chemical constituents or protein, there is Twinkle Mishra. You can raise hand, I will mute you. Hello, Twinkle Mishra, are you able to speak? She posted a message that it is a phytoconstituents, 3 phytoconstituents. So you are looking at protein or chemical constituent. Because the chemical constituent, then you
mute.
Yes.
Twinkle Mishra is still on mute. Or
Yes.
you have unmuted her.
I have unmuted her, but I don't know. So listen, that if you have a protein, you are talking in context of protein, then you can follow the things that design a network for pharmacology. But if you have go for a chemical constituents, then you just go for biogreen. you not able to test multiple chemical entities there. Just to check one and what are they. So that interactions of chemicals are pretty much difficult, and also the experiment very means no lots of experimental data is available which used to study for chemical interaction. So that is the issue. But you try with the biogreen. And protein, you can go with this. Anyone else? Okay, Twinkle. Now
Yes,
you are
sir.
on.
Now, yes.
Yes, you
Yes,
can.
am I
Yes, yes.
Yes, sir, I got your answer. But yes, that actually my question was that if there is some phytoconstituent like tripla formulations which we have, and we have to construct a network pharmacology for that. So, uh, so I was talking about that context only. Thank you, sir.
Can you repeat? Means phytoconstituent you
Yeah,
are
yeah, yeah. Sir. She was talking about Ayurvedic formulation like Triphala. So
talking.
Yes,
obviously,
yes.
then you have to use the other one. Yeah, sir, please continue.
Biogrid, and there is a but there is a no option to add the multiple. Suppose you have 10 compounds, okay? You want to see that because if you are doing phytoconstituents, means mixture of some plant powders and so on. So there are 10 compounds in one, 10 in other, and 10 in other, and you want to see. But the option is not available that multiple chemical entries you can do there. But one by one you can see, and if they are interacting with others, then automatically they will come in your result, and you can confirm that.
Okay, okay. So, sir, means in that if we want to connect our bioconstituent with targets and then with the diseases, so is that possible in cytoscape?
No. Cytoscape is only you use to analyze a network. You cannot go for its target, means how they are interacting or which phytoconstituent interact with which protein and so on.
Okay,
You only identify, suppose thousands of protein, with 1,000 protein you have started of a particular organism, and you are interested to know a particular protein molecule. So this ask you just to perform using the cytoscape to narrow down, because today is the era of big data, and in the proteomics we get thousands of protein, and we get the thousands of protein, but what is the main protein or what main protein we need to target? So you just go with this way and identify which proteins are we need to target to kill that organism and so on. Okay. Anything else? I think you feel that something is pending out of the list, or you wish to know about. Okay, then if no more questions and so on, can we stop today? You can raise the hand to say,
Yeah.
okay. Okay, then we will stop, and if anyone have difficulty, I will share my mail ID is there on the flyer, so you can contact me through mail if you have any additional information required from my end related your means you, if suppose you are doing particular work related to the protein, then you can feel free to come. So with this, I think we will stop today, and we with new enthusiasm, we will meet tomorrow for our next session on molecular model. Okay.
Okay. Thank you, Sagar sir. It was a very grilling day for you, and I hope the same is on the participants as well. So this sets the platform for tomorrow. Now, tomorrow we will exactly do the next step. Today we have um, completely focused on target identification, different proteins, receptors, and related aspects, how to shortlist the receptors. Uh, I would say a receptor from the homologous or functionally homologous proteins. It was a very insightful session. I would request the participants to try such sample data and try to execute the queries. Uh, so thank you everyone for the participation. Sir, what I will request is for day one, we can request participants to start the camera for one photo for
Yes, we
documentation.
will take.
I request
Actually,
participants can start the camera. Do they have the permission?
No, sir.
Okay.
so 1 minute,
Okay.
I will also, no. Sir, we can give now for time being, because tomorrow we
Sir,
don't.
sir,
Yes,
we
please.
need to, promote in panelists then,
Ah,
all of them.
No, 2, 3, or those who are
okay. I think
interested to.
those panelists who are there can start the camera.
Those who are interested, they can raise hand. Oh, we can 2, 3. We will convert. So, sir, you can convert. Means them as panelists, and tomorrow we will, or tomorrow or after this, we will again convert back to
Or else,
attendee.
sir, what we will do is tomorrow only we'll convert them. At the end of the session, we'll have a complete
No,
photo.
what happened?
Why Sitish is raising hand? Yes, Sitish.
So for that only, I thought, yeah.
What
Okay,
today?
for starting the camera.
Yes, sir.
Those who are converted as panelists, please start your camera. Aman Raj. Sir, are you able to, Kshitij, and are you able to on your camera? Switch on your camera, or it is not it?
Sir,
Sure.
support them in panelists, sir. You can see in more button, sir.
Okay. I will convert them. Promote to panelist. Okay. Promote to panelist. Error code it is showing. Only one candidate. I think only who are from
Gaurav Kadam is
Amity
there.
ID. Okay. Who are from
Ah,
Amity ID able to become a panelist. Okay. Aman Raj. Aman Raj, I have done already. Okay. So, yes.
Yes, sir. I can hear you.
You
yeah.
can switch on your
Kshitij
camera.
also started camera. Yeah, good.
Tomorrow also we will do.
Yeah.
Okay, we
Okay.
uh,
So this will, after this photo, will have a conclusion of day 1.
uh, okay, yeah. So we need to send them back in the attendees.
Yeah. We need to send them back to you.
Done.
Done, sir.
Done, sir.
So I will send them back. More. What to do, sir? Change role to attendee.
Sir, so I had a feedback. If
Yeah. Please
Yes,
tell.
yes, yes.
So it was a really informative session wherein I personally enjoyed because I was doing each and everything sir was doing parallelly. So it was a great session. I really liked the way all the instructors were getting us through all the content. Yes, thank you very much for this session. Looking forward to tomorrow's session.
Thank you, Satish.
Okay, we will meet tomorrow.
Yeah.
I will convert them in attendees. Still, Kshitij and Aman is there, right? As
Aman is there. I have
Aman, you
done.
don't want to become
Aman I did. Done, sir.
done.
Yeah, done. I
Aman,
think it is just showing
you
there.
want to become a panelist? Nahi, he is still there as a panelist.
Actually, I did, but he's not going. He's a panelist. Okay, let's see tomorrow. Or we'll request.
Rejoining as yes. Okay.
Yeah.
Okay, thank you all. And we will end today's session.
Okay,
Okay,
so,
sir.
as per the schedule, we will start tomorrow. Be 5 minutes before time, so that we will start exactly at time. I think tomorrow's session starts at 10 o'clock.
Okay.
Yeah.
Okay.
So we will start connecting for 9:50 or 9:55. Everyone will be in. We will start at 10.
Okay. Okay. Sir.
Okay, Sagar sir, thanks a lot.
Okay.
Thank you, everybody.
We'll leave, sir. Please end the meeting.
Yeah.